Greetings everyone. My name is Ndip Rita Ayuk from University of Buea, Cameroon, and I'll be presenting my work on a published article entitled Promising Activity of Tetrahydroisoquinolines Against Multidrug-Resistant Clinical Salmonella Isolates and Cytotoxicity Against Monkey Kidney Epithelial Cells under the supervisions of Professor Gogomo and Dr. Ngemeya. The outline for this presentation will run from a brief introduction through materials and methods to conclusion. Salmonella infections are caused by several serovars of Salmonella, typhi, paratyphi A, B, and C, typhimurium, and enteritidis. These infections could be transmitted through consumption of contaminated food or water. It's estimated that about 11 to 21 million cases, resulting to about 128,000 deaths occur worldwide. Figure 1 shows a distribution of these infections worldwide, in which we see regions in red indicating high incidences, region in blue indicating medium and low sporadic cases found in developed countries, and this could be due to the differences in sanitation measures and hygienic measures put in place as well. In Cameroon, several reports have been given on the prevalence of these infections. In 2019, a study in Buea Health District reported 27.3%, in Littoral 32.5%, and in the Northwest region 30.1%. All these areas are found in Cameroon, and this is just to show that this is a problem of public health concern in our region of study. The treatment for these infections include chloramphenicol, ampicillin, and trimethoprim sulfamethoxazole, which were the first-line drugs of choice in the 1990s, in the 1940s, and due to the emergence of resistance to this infection against antibiotics, fluoroquinolones and the third-generation cephalosporins were introduced. And to date, the World Health Organization has recommended that treatment of these resistant infections be done by use of azithromycin and the carbapenems and aztreonam. Antimicrobial resistance occurs when a drug of choice or an antibiotic is unable to treat an infection due to a resistance or due to the consumption of these antibiotics misuse. That's when we hear cases of antimicrobial resistance. And here in Cameroon and the Central African sub-region, several reports of multi-drug-resistant strains of Salmonella have been detected. And also there is genetic resistance linked to fluoroquinolones. And there we have, as a result, decreased susceptibility and treatment failure. To this effect, we have several approaches which are being used in order to discover new antimicrobials. We have chemical modification from existing antibiotics, and this could be done by chemists using several methods. We equally have synthesis of new antibiotics, which could be from a rational drug design whereby we have a target, and from several molecules we can form new drugs and use them to test against other diseases so as to know if they can be used to combat. We equally have combination therapy whereby two or more drugs are used to treat a disease. We equally have screening of synthetic compounds from medicinal chemistry. Like in the case of this study, it's our main focus here in drug discovery. And for this, tetrahydroisoquinolines, which occur widely in nature and possess bioactivity to several microorganisms, be it viruses, bacteria, and parasites. These compounds have heterocyclic nitrogen in their basic scaffold, and they've been shown to have activities to other microorganisms. Figure 2 shows the synthetic schemes of these compounds. 1-aryl-6,7-dihydroxy 1,2,3,4 tetrahydroisoquinolines, synthesized by Gohana et al. And these compounds have been used to test against parasites, the malaria parasite to be precise, and it was shown to have some activity. The rationale for this work, due to the fact that Salmonella infections cause a problem in a region of study, it causes high level of morbidity and mortality in humans, and this also has been aggravated due to the spread of multi-drug-resistant strains to all recommended antibiotic classes. This therefore justifies the reason why there is the search for a new efficacious anti-Salmonella agent. Due to the fact that several reports of tetrahydroisoquinolines have shown broad-range bioactivity, we thought and thought it wise that we could use this in order to check for their activity against multi-drug-resistant Salmonella strains. As objectives, the general objective for this work was to screen a series of 17 tetrahydroisoquinolines against multi-drug clinical Salmonella isolates, specifically to assess the anti-Salmonella activity, and 2, to assess the toxicity of the active tetrahydroisoquinolines on monkey kidney epithelial cells. Clinical isolates were obtained from health facilities around Southwest region Cameroon, basically hospitals which carry out culture, and anti-susceptibility testing was done on some antibiotics by method of Ayuk et al. Anti-Salmonella screening of these compounds were done by Dix diffusion, whereby McFarland standards of 0.5 were prepared, spread on a plate, and the compounds impregnated in Dix and placed on the surface, incubated at 37 degrees Celsius, and thereafter the zone of inhibition was measured so as to get the inhibition of growth, followed by microdilution method, in which the minimum inhibitory concentrations and minimum bactericidal concentrations were obtained. The cytotoxicity assay was also done on monkey kidney epithelial cells, where 50% of cells were assessed, and for data analysis we used measurement of diameter, zone of inhibition, and minimum inhibitory concentrations, as well as graph path prisms were done. For interpretation, high activity was greater than 20 millimeters, moderate 15 to 19 millimeters, and weak activity between 7 to 14 millimeters. That's for the Dix diffusion. And for the microbroad dilution, high activity was less than or equal to 4 microgram per mil, moderate activity 8 to 16 microgram per mil, and weak activity greater than 32. For the results and discussions, figure 4 shows some plates in which we can see, compared to the positive control used, which was chloramphenicol, we had some visible and clear zones of inhibition. If we take the case of compound 2 and 14, we can see on all plates a very clear zone of inhibition, among others. Table 1 shows the Dix diffusion screen that was done, and we had compounds 2 and 14 giving very high activity, and it falls within the moderate range as we earlier indicated, followed by two other compounds, compounds 3 and compounds 15, compared to the positive control, and we went further. For the micro dilution assay, we had MIC, MBC, and MBC-MIC ratios for two compounds that were most active, compound 2 and 14. As we can see, we noticed that they gave the highest number of MIC and MBC and MIC ratios, indicating that these compounds are bactericidal. Compounds 3, 4, 5, and 17 equally gave some MIC values but had low MBC values recorded. As you can see, their MBC-MIC ratios are not indicated, making them weak. If we look at the results from the Dix diffusion and the microdilution assays, we see that there is a trend of activity indicating that those compounds were indeed having moderate activity, and it's promising to do further research on them. For the cytotoxicity assay, we did an analysis based on the lowest MIC value that was obtained, and the two most active compounds, compounds 2 and 14, we had compound 14 giving a very high CC50 value, whereas compound 2 gave a very low CC50 value. Other compounds that showed some good CC50 values were compounds 3, compounds 4, compounds 15. And with this, we can infer that these compounds have shown that they possess low toxicity on monkey kidney epithelial cells. As conclusion, the 17 tetrahydroisoquinolines studied have demonstrated a promising but structure-related antibacterial activity against multidrug-resistant Salmonella. Therefore, medicinal chemistry exploration may yield active antibacterial lead. Based on the cytotoxicity tests, it indicates that these compounds may possess low risks of toxicity. Therefore, chemical modification of this series should be done to increase potency while reducing toxicity. And we can see that the work was published in the Scientific African Journal in 2022. Some references. I would like to acknowledge these persons, the collaborators from the Chemistry Department of Faculty of Science, University of Buea, for providing all synthetic compounds, the funders, African-German Network of Excellence in Science, for the synthesis of these compounds, the Biotechnology Unit of Faculty of Science for access to research facilities, the Eurasia Research Conference team for giving me the opportunity to present my work. Thanks for listening.